60 to 90% chance of also having a disorder on the autism spectrum. So we know that genes play a big role and that there's a large amount of heritability defined by that sort of twin study. Although we don't yet know what all of those genes are. We've made a lot of progress, much of that spearheaded by the support of the Simons Foundation, in the last 10 years or so in understanding a lot about the genetics of autism spectrum disorders. But as I say, there's still a lot that we don't yet know. So autism can be represented as a pie chart. This is if a child comes into the clinic and we try to do a bunch of genetic diagnostic tests to try to figure out what is causing the autism in this child. Our chances of finding an answer might be something like 1 in 3 or 1 in 4, because about a quarter of the pie chart of the postulated causation or the expected causation of autism is presently discoverable by the technology that we have. And the most important technology for understanding genetic causes of autism has been a technique called whole exome sequencing, which is essentially like whole genome sequencing, where you sequence the entire genome, but the exome sequence is just that portion of the genome which actually codes for proteins. It's only about 1% of the genome, and it's faster and cheaper to sequence. So a lot of whole exome sequencing has been done, and that has really informed us about what causes autism. So there are a variety of single gene disorders that can be associated with autistic symptoms that might be familiar to some of you. Conditions such as Fragile X syndrome or tuberous sclerosis syndrome. And these have been known for many decades as having autism as some of the symptoms, but they also cause other things. Tuberous sclerosis, for example, causes tumors, and so it's relatively easy to recognize. And collectively, these Mendelian or genetic disorders cause a few percent of the condition. Some kids have chromosome abnormalities where their chromosomes are rearranged or broken or translocated in ways that can be recognized by looking at the chromosomes under the microscope, but that's relatively rare. And then with the advent of the sequencing of the genome, that allowed us to look at the whole genome. And starting about 10 years ago, there was a recognition that copy number variants in the genome were a very important cause of autism spectrum disorder. And for those of you who aren't familiar with copy number variants, this refers to either a deletion of part of the genome or a duplication of part of the genome. So that instead of the usual 2 copies that we have for most of our genes, a copy number variant might be that one has 3 copies or else 1 copy for some segment of the genome. And these copy number variants tend to be at about 1 million base pairs or so in size. And they can occur in a variety of places in the genome. And collectively, they can cause autism. There are a few recurrent copy number variants, such as deletions of chromosome 16p11 or deletions of chromosome 22q11, but most of the copy number variants are not recurrent, and they can involve a variety of genes throughout the genome. And then using whole exome sequencing, more recently, people recognized that you could have, and these copy number variants are typically de novo, which means that the parents are normal, and there was, in the copying of the DNA in the sperm or the egg, basically a segment of the genome was either lost or duplicated. And so that's why we call them de novo copy number variants, meaning present in the child and not present in the parent. So with the sequencing of the exome, we find that there are also other kinds of de novo mutations, but these are point mutations, where just a single base out of the 3 billion base pairs of our genome is changed from one base to another. And that single base pair change, if it occurs in exactly the wrong place, can be enough to disable one of the 2 copies of a gene. And so these are called de novo point mutations, meaning a single base pair mutation. And again, the ones that we are able to assess are present in the affected child, but not present in an unaffected sibling and not present in the parent. And again, they seem to occur during the copy of the DNA that occurs during the formation of the egg or the sperm. And then there are also a variety of recessively acting mutations. Recessive, and these tend to occur also in the coding region of the genome, the exons that actually code for proteins. Recessive mutations are conditions where the parents are silent carriers and the child is affected. Conditions like cystic fibrosis or various hemoglobinopathies like sickle cell anemia. But there are also recessive mutations that can affect the brain, and some of these manifest as autism, and they cause about 3% of the disease. So as you can see, there's still a lot of this pie chart left undiscovered. And so what other, and there's still a lot of, and these collectively account for maybe 25% or so of cases of autism. And so that doesn't account for all of what we know should be the genetic contribution of autism. Autism is not necessarily 100% genetic. We don't expect every child to have a genetic diagnosis, but we do expect the majority of children to have a genetic diagnosis ultimately. And so what is missing from this pie chart? So that, at the moment, is an unknown, to quote whoever that defense minister was. And especially, in fact, what we really don't understand is the high-functioning autistic children. Because most of the mutations I just told you about tend to cause not only disorders of social behavior that we refer to as autism, but typically disorders of intellectual behavior. And it's usually children with relatively low-functioning autism, which are the children where we can actually identify one of these mutations that I just told you about. So we really lack an explanation for the people like Kim Peek, who was this brilliant autistic savant who recently died, who was the model for the Dustin Hoffman character in The Rain Man. So he could do incredible feats of mathematics and memory, and yet he had horrible social skills. And this kind of autism is relatively rare, but we really don't have an understanding of whether it has a genetic cause, and if it does, what the genetic causes might be. And so we assume that these might be subtler types of mutations that are not picked up by exome sequencing or by looking for copy number variants. And there are various genetic mechanisms that might cause some of the rest of autism. There might be common variants in the genome that might predispose to risk, and I'm not really going to talk about that because that is probably an important mechanism, but not one that we've worked on. But I will actually talk about two different mechanisms that seem like they might be important, particularly for some of these rare cases. And really what I'm trying to understand is, how can you have a brain where part of the brain seems to work so beautifully well and other parts of it seem like it can work so poorly? And what can that tell us if we understood that about what is the anatomy of autism in the brain, or the mechanism of autism in the brain, and how might that help us understand the condition so that we might potentially treat it better? And so I'm going to talk about two mechanisms which both have the potential to allow part of the brain to work perfectly fine and the rest of the brain to be relatively disabled. And one of these mechanisms is called somatic mosaic mutations, and I'll talk about that in the first half of the talk. The other mechanism is non-coding mutations, and I'll talk about that in the second half of the talk. So what are somatic mutations? Somatic mutations really are a non-inherited genetic variability that lies within all of us. So I told you that these de novo mutations that can cause autism are caused when the DNA is replicated in the sperm or the egg. But every time a cell copies its DNA, it runs the risk of making an error. In fact, it reliably makes an error. There are about 100 mutations introduced every time a cell divides. And so it's kind of like making a photocopy where the photocopy is always degraded. And so somatic or mosaic mutations are mutations that occur during the mitotic cell divisions that generate our body. And so therefore, they're not present in all of the cells of the body, but they're present in some and not others. And somatic mutations are actually familiar because they're the cause of cancer. Mutations that arise in your lungs because of smoking eventually can cause a gene to be hyperactivated that can cause lung cancer, or similar mutations in the skin, if you get enough of them, can cause skin cancer. But only recently have we recognized that somatic mutations can occur in the brain as well and create a brain where some of the cells carry a mutation, but not all of the cells. And so what I'll tell you is that somatic mutations can cause epilepsy. They're typically missed by our usual genetic diagnostic procedures like whole exome sequencing or other clinical methods. And in fact, the genome of every single neuron in the brain is unique, although I'll only talk about that fairly briefly. And they're unique because every cell has its own list of somatic mutations. And in fact, these somatic mutations mark the development of the brain and represent a record of how the brain develops. So, as I said, genetic mutations cause disease, including neurological disease. And I was telling you about, and so this shows two types of mutations. This is an inherited mutation where the mother inherited a mutation, and the mutation goes into her eggs and is inherited in the zygote after fertilization, and is therefore present in all of the cells of the body. This represents the non-brain cells, and this represents the brain cells. And an example is an inherited epilepsy gene. This is a de novo mutation that had occurred in the sperm of the father, So the father does not carry the mutation. It has never appeared before in the family. And in fact, if you looked at the father's blood, you would not even see the mutation because it's only present in his sperm cells and only in some of them. But then it goes into the zygote, and so therefore the child derived from the zygote carries that mutation in all of the cells of the body. And that's a de novo mutation. An example is a mutation in a gene called LIS1, which if it's de novo, causes the whole brain to be horribly malformed instead of the normal folding pattern. And these kids have intractable epilepsy and typically die before they ever grow up old enough to have children themselves. And so the only way we ever see the disease is because of these de novo mutations, because this kind of mutation can never be inherited. These children can never have children of their own. Now, as I explained, somatic mutations occur also during the replication of DNA, but during mitosis. So not in the sperm or the egg, but during when these cells are dividing. And so in this case, this dividing cell here gives rise to some cells in the brain and also some cells out of the brain. And so this particular somatic mutation occurs relatively early and is present in about maybe a third of the cells of the body, but not all. And so if you look really carefully in the blood, you can probably detect it, but you have to look very, very carefully. And it causes a malformation of the brain, but a milder one, where the brain is sort of abnormally folded, but not quite as severely. And so these patients have a milder form of intellectual disability and epilepsy. And then I'll tell you also about somatic mutations that occur even later. And so they're only present in some of the cells of the brain, but they occur after the brain cells separate from the blood cells and the lung cells. And so you can only detect these mutations by studying DNA from the brain itself. And we've only recognized these recently. And an example is a mosaic mutation in a gene called AKT3, which is present in this half of the brain, the right side, which is shown on the left side by radiographic convention, but not present on the left side of the brain. And so it actually, not only does it occur after brain separates from blood, but it actually occurs after the right hemisphere of the brain separates from the left hemisphere of the brain. And so this is what that looks like. These somatic mosaic mutations that can be in half of your brain, they cause a condition that's called hemimegalencephaly. And just to decode that, hemi means half, mega means big, and cephaly means brain. So it just means half of your brain is way too big. And so this is what a normal brain looks like, sectioned up and down. Here it's sectioned parallel to the brim of a hat. And in this case, you can see that this half of the brain is a lot bigger than that half of the brain. And it's overgrown and it's disorganized and it doesn't show the normal signaling characteristics. And this child has intractable epilepsy arising from just that half of the brain. As far as we can tell, this half of the brain works perfectly fine. When this child was born, he started having seizures right at the time of birth. He never developed language. The left side of his body, which is governed by the right side of the brain, was weak because this big malformed brain doesn't work properly. And so his seizures were so intractable that he had no hope for a normal life. And so his neurosurgeons removed his entire right hemisphere. And so this is just water where half of the brain used to be. And for this child, this was life-saving. He had no seizures for 6 years. He learned to walk. He developed fluent speech. He's remained weak on the left side, although it mostly affects his hand. He now is at school at grade level. And this is work, by the way, of Ann Podori, a child neurologist with whom I collaborated on this study. Here's a picture of Dante, whose name and picture I used with his parents' permission, and he would tell you he's a perfectly normal young boy. His parents recognize that this left arm is quite weak, but he's doing amazingly well, considering the deficits he had and the challenges he's had. And so Anne collected 8 cases of hemimegalencephaly, and we assumed that this was going to be due to a mutation, like a cancer mutation, that's in this part of the brain and not in that other part of the brain, and we assumed it probably would not be detectable in the blood either. And to summarize a few years of searching, almost a decade of searching, she identified in Dante a mutation in this gene AKT3. And in fact, this is what the blood looks like, which is a normal sequence of TA, GG, AGA, and so on. And in Dante's brain, there's just a little extra peak that you could barely see unless you're really, really looking for it. And it's only present in the brain and not the blood. And this is actually a known mutation which has been previously associated with certain cancers. This substitution of glutamine at position 17 for a lysine, it activates cells to overdivide and prevents their normal differentiation, and that's why his brain is too big and the cells are all so abnormal. And that you absolutely cannot detect this mutation in the blood because it only arose in the progenitors for the brain. And other work from several other labs, including the Baulock lab, the Biesecker lab, Bill Dobbins, Joe Gleason, the Lee lab, Ingrid Scheffer's lab, all at about the same time, discovered that hemimegalencephaly is actually just one of a variety of overgrowth syndromes where you can have this spontaneous activation of this growth pathway due to these somatic mosaic mutations. And the genes associated with these overgrowth syndromes all focus on a single biochemical pathway known as the mTOR pathway, the mammalian target of a drug called rapamycin. And this regulates growth factor signaling and cell growth throughout the body. And if you happen to have these mutations in your foot, then you get overgrowth of the foot. If you happen to have them in your big toe, you get overgrowth of your big toe. If you happen to have the same mutation occurring in your blood vessels, you'll get overgrowth of blood vessels. And if you have it in your brain, you'll have overgrowth of your brain. And you can have activating mutations of PI3 kinase, or you can have activating mutations of AKT, or you can have activating mutations of mTOR itself. Or you can have loss of function mutations of a variety of proteins that normally inhibit this pathway. And so if you basically destroy the brakes or if you slam the gas to the pedal floor, you have the same effect of uncontrolled overgrowth. Now, it's not completely uncontrolled because in the brain, these kids don't get tumors. They have a stable overgrowth, but I guess because the neurons don't normally divide, they still stop dividing. And so typically, hemimegalencephaly is not associated with tumors, but more of a stable overgrowth. And that's a whole other story why that might be. Now, you can also get the exact same kind of hemimegalencephaly by loss of these negative regulators. And an example was also found by Anne and a student, Alyssa DeGamma, where they studied other cases of hemimegalencephaly, and they found that the patient inherited a mutation in a gene called tuberous sclerosis 2, but then just in the lesion, there was only about 5 or 10% of the cells had a loss of the second copy of this gene. So there are normally 2 copies of this gene, and if you inherit one deficient copy and then you lose the second copy, just the cells that lack both copies will have this overgrowth and this hemimegalencephaly. And we had a couple of different cases of that, and actually, surprisingly, neither of these patients had other manifestations of tuberous sclerosis. They both had hemimegalencephaly, just this very focal overgrowth. And so this actually shows how you can have an interaction of these somatic mutations with the inherited DNA that you inherit from your parents. And sometimes it's not just the somatic mutations or the inherited mutations, but the combination of the two. Now, there's another overgrowth syndrome that has certain resemblances to hemimegalencephaly, but is a little bit milder, and that's known as focal cortical dysplasia. And this is a very small overgrowth of just a little segment of the cortex, this little bright white spot here, or this little spot right there, or this little spot here, or this little spot here. So focal cortical dysplasias are lesions that are only about 1 centimeter or 2 in size, and they can occur anywhere in the cortex. And they also cause intractable epilepsy of children. In fact, they are the most common cause of intractable epilepsy in children. And they also respond very well to surgery. So neurosurgeons love to take these out because they typically, in 70% of the cases or so, will cure the epilepsy. And they are due to the exact same mTOR pathway mutations. They are only present in that tiny lesion, and they're typically only present in less than 10% of the cells of that lesion. So not even all the cells in this lesion carry the mutation. And so these obviously arose long after the brain cells separated from the blood cells. And so it's only by studying the tissue that you get at the time of surgery that we could actually even discover that these mutations are present. And so it's the timing of this mTOR mutation that causes the lesion. And here's an example. This is work of Alyssa DeGamma and Ann Podori. So here's a case of a focal cortical dysplasia. There's just a little lesion here where the gyri don't look quite right, and you have to be sort of an expert to see that. And this particular lesion, if you take it out and study it in the lab, you'll find that about 12% of the cells have a gain of function mutation in mTOR itself, the cysteine 1483 to arginine. and that's another activating mutation. And as I say, it's only present in about an eighth of the cells. And then almost the exact same mutation, this time a cysteine at the same position to a tyrosine, causes a similar activation. In this patient, it's present in about 20% of the cells throughout a whole hemisphere. And so that's why this patient has hemimegalencephaly instead of a focal cortical dysplasia. And then here's another child who has the exact same mutation again, but in this case, it's a de novo germline mutation. And in this child, the entire brain is too big. In fact, there are other lesions outside of the brain, and this child had intractable epilepsy and died in the ICU before the age of 3. And so it's not just the type of mutation, but also the timing of when and where it takes place that defines the sort of disease that these will cause. Here's another example, an FCD with another mutation here, only present in 3% of the cells. Another FCD that has the exact same mutation present in 3% of the cells and not outside of the brain. And then again, the same mutation in a larger fraction of the cells. It ends up spreading over a larger part of the brain and enlarging in a whole hemisphere. So the most common cause of intractable epilepsy in children is due to a somatic mutation that's only detectable in the brain, and in fact only in part of the brain. And so these are not necessarily rare conditions. And so we wanted to know whether there might be cases of autism spectrum disorders that might have this same sort of, it might be the same sort of distribution of a mutation, maybe present in half the brain and not the other half of the brain, but in a different gene, not in an mTOR gene that would cause overgrowth, but maybe in one of the many known autism spectrum genes. So this is just reviews how we define autism due to its impairments in social spheres. As I say, the majority of children with autism have cognitive impairment as well, and seizures are not uncommon. And as I say, it's very highly heritable. So just to review the chart here. So we've approached somatic mosaic mutations in 2 ways. One way is to look for somatic mosaic mutations that might be present in the blood as well as in the brain. And by looking at large numbers of exome sequences, we looked to see whether we might detect certain mutations in a small fraction of cells. And this is work of Elaine Lim. And we looked at more than 5,500 exome trios. And what she found is that if you look at the de novo mutations, most of them have a alternate allele frequency or a mutation frequency of 50%, and that would be consistent with it being a mutation that occurred in the germline. But that there's this significant peak here of de novo mutations that seem to be only in a fraction of the cells, and that would be consistent with them having arisen a little bit later and not being in the germline, but being, again, in some of the cells and not others. In order to identify these better, she developed a whole new calling algorithm. She tested it repeatedly by doing a series of validation experiments to test and to confirm whether the predicted somatic mosaic mutations were actually real. And in fact, found that the majority of the somatic mosaic mutations she identified had never been found before because other calling algorithms would miss them. And overall, she found that about, after rounds of confirmation, we find that about 7% of the de novo mutations are somatic mosaic mutations that occur early enough to be found in the blood. We know we will be missing somatic mosaic mutations that are only in the brain, and so we're only capturing a portion of them. And so they represent a small fragment, a small piece of the de novo mutations overall, but they are common enough that they might account for some of these higher functioning patients. And in fact, if you look at the IQs of patients that have these de novo somatic mosaic mutations, the numbers are small, but in fact they have significantly higher IQs than patients that have germline de novo mutations. The other way then to look for somatic mosaic mutations is to study the postmortem brain of a child who died who had autism spectrum disorders and directly sequence the DNA to see if we might find mutations that might be only detectable in the brain or only detectable in part of the brain. Now with autism compared to hemimegalencephaly, we don't know where to look. Here we know where the mutation's going to be. With autism, we don't know. We just have to take our best guess. And so we just Alyssa decided to sequence DNA from the frontal lobe of autism, since that's been repetitively implicated as a potential location for autism spectrum disorders. And also here we only had about 50 autism brains to work with. And again, thanks to the Simons Foundation and others for doing a very good job to try to organize and increase that supply. And what she did was then sequence the best known autism genes, about 75 in all, at very high coverage, so that we would have very high sensitivity to detect a somatic mosaic mutation, even if it was only present in a couple of % of the cells. And so what we found was we sequenced 50 or so cases and 50 controls. We found that when you compared the two, the cases, shown here in red, had a much higher rate in which we would find a potential disease-associated mutation. And in fact, if you looked at the most severely deleterious mutations, in fact, there was more than a twofold excess in the cases compared to the controls. And if you looked at the most severe loss-of-function mutations, they were only seen in the brain of autism cases and not in controls. And so we identified in this way that a lot of these brains, actually, of kids had it didn't just have autism, but they actually had autism from a definable genetic cause, which we can now use to understand more about potential mechanisms of what happens in the brain of patients that have particular genetic causes of autism. We were amazed to find that some of the well-known causes of autism were seen repeatedly even in this small sample. So SCN2A, one of the best-known genes, showed 2 loss-of-function mutations and 2 very likely deleterious missense mutations in 4 different cases. And SCN1A was recurrent, and ARID1B was recurrent as well. And in many cases, when we looked back on the charts of these patients, we could see very strong clinical evidence that suggested that our genetic diagnosis was correct. One patient had a PTEN loss-of-function mutation, and that's known to be associated with a brain enlargement because PTEN is one of those negative regulators of mTOR. And in fact, this patient was known to have had a large brain and epilepsy, consistent with this being a deregulation of mTOR. Another patient had an SCN1A missense mutation and had a family history of epilepsy that's very consistent with that diagnosis and so on. So we found a couple of these de novo mutations were, in fact, somatic mosaic mutations, but we only found a couple. So the small numbers do not allow us to show that they're more common in cases than controls. They're nonetheless intriguing. There's one, a de novo mutation in SETD2 is interesting because we only found it in the cortex, shown here in blue, and the mutation is undetectable in the cerebellum. And so you can see that here. The mutation here is present in about a quarter or so of the cells in the frontal lobe, but not in the cerebellum, and we think it's probably only present in one of the 2 hemispheres, analogous to a hemimegalencephaly mutation. Or another somatic mosaic mutation here is a clear loss-of-function mutation in SCN1A that disrupts a splice site. This one was present throughout the brain and a much larger fraction of the cells, actually almost 90% of the cells. And so that must have occurred very early, probably at the 2-cell stage. I'll just build this whole thing. And so then this shows those deleterious mutations. There was another somatic mosaic mutation that was present in the frontal lobe and in the cerebellum. And this was a mutation that destroyed the start site. So it was a complete loss of function in a gene called calcium channel gene. This was found actually in the brain of a patient who carried the diagnosis of Fragile X and clearly had a Fragile X mutation. So this patient had 2 mutations, the germline Fragile X mutation as well as a somatic mosaic mutation. We found 2 patients that had a Fragile X pre-mutation in their blood. Not the full Fragile X expansion, but only a pre-mutation. But then when we looked in their brain, we found that there was an expansion of the Fragile X repeat in the brain only, so that the Fragile X gene would be inactivated in parts of the brain. In one case, both in the cortex and the cerebellum, and in another case, in the cortex, it was inactivated, but in the cerebellum, in fact, it was not expanded, suggesting that the Fragile X mutation is actually dynamic in different parts of the brain. And so, as I say, we have relatively small numbers of these somatic mosaic mutations, but as we accumulate more of them, we hope that we might be able to study their differential distribution to ultimately understand a little bit more about the localization of autism spectrum disorders. We found also a couple of, this is a synonymous mutation, this is an intronic mutation, these are probably non-functional. In other words, they probably do not impair the function of the gene. We assume that they're silent and that the gene is still okay. But they also show how these mosaic mutations can be, in this case, present in the cortex and not in the cerebellum, or in this case, present in the cerebellum and not in the cortex. So what we've been doing recently is doing 200x whole genome sequencing on patients' post-mortem brains with autism, because this will allow us not just to study those 75 autism genes, but to look at the entire genome. And this will allow us to look at copy number variants and single nucleotide variants and everything. And we've just collected that data. We haven't analyzed it yet. But this just shows something early. One of the patients turns out to have a deletion of Nerexin-1, which is a known causative mutation in autism spectrum disorders. That appears to be a germline deletion, but at least one other patient looks like it has a potential somatic copy number variant as well. So the time and the place of mutation looks like it's critical for determining its consequence of the mutation. You can have germline mutations present in all of the cells, or you can have these somatic mosaic mutations that might be present throughout the brain, even though they occurred slightly after fertilization. Or you can have mutations like this SETD2 mutation that looks like it's in half the brain and not in the other half, and only present in about half the cells. And that's very similar to the picture where if you had the gain-of-function mTOR mutation, that would cause hemimegaloencephaly. And then we also have these focal cortical dysplasia mutations that can impair just a tiny segment of the brain. We have not yet found anything like that for autism mutations, but you can envision how you might potentially have mutations like this that might only impair a portion of the brain. So somatic mosaic mutations cause brain disease. The question is just how common is that, and what's the range of diseases that are caused by somatic mosaic mutations? As I say, you can have mutation of as few as 1% of the cells in a tiny portion of the cortex, and that can cause intractable epilepsy. The topographic extent and presented mosaicism relates to the timing during development. And as I say, we have some preliminary evidence for a role in autism spectrum disorders as well. And we don't know the extent to which these somatic mosaic mutations, we know that they occur broadly in the normal brain. We don't know to what extent they may or may not shape the development of our brain. And I think I'm going to skip this part, actually, because we have a whole study of the normal patterns of somatic mosaic mutations. But I don't want to run out of time, and I want to make sure I have enough time for the second part of the talk, or the last third of the talk, I should say. What are other genetic causes of autism spectrum disorders that might be missed by whole exome sequencing? The last part of the talk, I'll tell you about mutations outside of the exome in the non-coding parts of the genome. And again, this is a study that would not have been possible without the generous support of Jim and Marilyn Simons, who allowed us to collect a unique cohort of patients on the autism spectrum from Middle Eastern countries where marriage between cousins is conventional. And so you see that in all of the Arabic countries of the Middle East, marriage between cousins occurs more than 10% of the time, and in some countries like Saudi Arabia, it occurs more than half of the time. And so it's the typical. And so we thought that we had known for years that uncovering recessive mutations, where you have to have a double dose of the mutation to cause the disease, is always easiest in these consanguineous populations that share common ancestry, because in these cases, the parents tend to carry the same recessive mutations and are more likely to have an affected child. And so we thought they might allow us to also uncover the recessive mutations that contribute to autism spectrum disorders. So the first thing we did was sequence the exomes of these patients, and we found that some of them had coding region mutations that were recessive, but in fact a surprisingly small number of them. The other thing we looked at was to look for copy number variants. And these families have the same sort of copy number variants that American kids can have. So this is a de novo loss of one copy. You see how the pink here is paler because one of two copies of this DNA this is basically genotyping of a chromosome. And so there's genotypes missing here because it's paler pink. And that represents loss of one of two copies of the entire segment of the chromosome. But these consanguineous, and these are typically large and contain many, many genes. But these consanguineous families have a second kind of copy number variant that you almost never see in America, and that's an inherited one, where the father, shown by the square, has this small deletion here. The mother shares the same exact deletion. The unaffected sister has the same exact deletion, but the son is completely missing DNA altogether. And so we call this a copy number 0 or a CNV0, meaning it's a complete deletion of both copies of a portion of the genome. And we reported a couple of years ago that these seem to be common in these consanguineous families. They tend to be much smaller, and they typically involve anywhere from 0 to only a couple of genes. And so most recently, Klaus Schmitz-Abe and Kyriakos Markianos have studied these in more detail. And so this is a figure from a paper about the Simon simplex collection showing that these de novo heterozygous copy number variants are about 3 times as common in cases compared to controls, either the duplications shown in blue or deletions shown in red. When we do the same thing, when we look at a highly consanguineous family, in fact, the affected individuals don't actually show any more de novo copy number variants than unaffected ones. But if you look at families that happen to live in the Middle East but where the parents are less closely related, you see a similar excess in cases compared to controls, although the small number of patients makes the difference not technically statistically significant. On the other hand, if you look in America, you almost never see these CNV zeros. In the Simon simplex collection, they're equally rare in cases and controls. In the AGREE, which is made up of families with more than one affected child, they seem to be slightly more common in cases and controls. But if you look, and then if you look at Middle Eastern families where the parents are not particularly closely related, they're equally common in cases and controls. But in fact, the families where the parents have close relationship, like a cousin marriage, they're about 5 times as common in affected individuals as they are in unaffected individuals, strongly suggesting that the great majority of these, perhaps 75% of them, contribute at least somehow to the risk of autism in those kids. And now, surprisingly, we thought these CNV zeros would usually just delete a gene and cause autism that way, but in fact they don't usually. 10 out of the 28 that we've identified delete part of a gene or all of a gene, but in fact the majority of them delete specific non-coding parts of the genome that have been previously recognized to have regulatory sequences like promoters and enhancers. And this is just 3 examples. So here's a deletion that's near a gene called SCN7A that's expressed in the hypothalamus and regulates the secretion of oxytocin and vasopressin. And here's a homozygous deletion that leaves the exons shown here alone, but deletes the DNA right next to the start site of the gene, and that includes the promoter, which is marked by this H3 histone 3 lysine 4 trimethylation, which is a mark of promoters, and it also deletes, and you can see that it's a promoter in neurons as well. So in fact, the gene's still there, but it doesn't look like it's working quite properly because it's missing some of the on-off switches that would normally regulate its expression. Or here's another patient that has a deletion here, which seems like it's in the middle of nowhere. The nearest gene is ERS1 all the way over here. But in fact, there's a technology that can connect regulatory sequences to their target genes, and it's called circular chromosome conformation capture, because what happens is enhancers that regulate genes actually connect to them as part of a complex. The DNA bends so that the enhancer is brought next to the gene body and the promoter, and if you actually precipitate this complex and then cut the DNA and religate it and sequence it, you can actually connect enhancers to genes. And so we know that this enhancer here is actually connected directly to the promoter of that gene and regulates that gene. Well, here's another homozygous deletion, and again, this is an enhancer, a regulatory sequence marked by this histone 3K4 monomethylation, and this has been connected to a gene called UNC5, which is a receptor for netrin, and which is highly expressed in the developing neurons of the cortex, and in fact in the postmitotic neurons of the cortex as well. And so we think that the gene is there, but it's just not working properly, and that impairment then might cause the autism spectrum disorders. So in the last few minutes, I'll tell you about other non-coding elements that show mutations in patients with autism spectrum disorders. And again, these non-coding elements are also mostly enhancers. But this is a particularly interesting set of non-coding elements in the genome because these are elements of the genome that have previously been suggested to regulate the evolution of humans, and specifically the evolution of the brain. And these sequences are called human accelerated regions. And there are about 2,700 of these regions in the genome. Each segment is maybe only a couple of hundred base pairs long. And they're non-coding segments. They usually seem to function as enhancer elements that regulate some nearby gene. And they're defined by the fact that they're highly conserved in many, many mammals. So here's a sequence of a HAR. And the dot represents an identical sequence in chimps and gorillas and orangutans. So this sequence is highly identical in many non-human species. And whenever geneticists see this sort of conservation between species, it's a mark that it must be doing something really important, which is why nature left it alone. And then when you look at the human, it's mostly similar, but it's just different a little bit. And that suggests that it regulates a conserved function, but a conserved function that's a little bit different in humans than other animals. And so these can be highlighted throughout the genome as potential locations that might actually regulate the evolution of humans. And there are many of them, and we really, with 2,700 of them, no one knows where to begin, which ones are the likely to be important ones, and which ones might just have been found by chance. And so we wanted to ask, do mutations in these HARs, do they cause social and cognitive disorders, things like intellectual disability and autism spectrum disorders? And so can we try to identify an essential set of HARs because the social behavior of non-humans is so different than the social behavior of humans, where we live in great big packed communities like New York City. Is there an extent to which this difference in behavior is encoded in some way in our genome? Is it all cultural, or are there genetic elements to it? And so we thought if there are HARs that play some role in social or intellectual evolution, if you completely disable that HAR or destroy it or severely change its function, it might cause disorders of the social or intellectual behavior. So as I say, HARs are usually non-coding, and they frequently show the marks of enhancers. So 60% of them are between genes. 30% of them are between exons of genes and the introns. And again, if you look at these marks of enhancers as H3K4 monomethylation, especially fetal brain enhancers, that's significantly more common in HARs than in the genome in general, particularly the fetal brain enhancers. And other HARs are probably enhancers in other tissues. They're not all just brain ones, but we wanted to focus on the neural HARs, or the HARs that are likely to have functions in the brain. And as I say, we wanted to do several things with these HARs. We wanted to try to figure out not only do they carry mutations, but if they do, what gene does that HAR regulate using this chromosome capture technology. And this is work of Ryan Doan, a postdoctoral fellow in the lab. So going back to the mutations that we know cause autism, we wanted to explore two types of mutations, but focusing on whether these mutations might disrupt HARs. So we looked at de novo copy number variants that involve HARs, and we looked at recessive mutations in HARs. And so for the de novo CNVs, we went back to the Simon Simplex collection and asked what proportion of these de novo CNVs involve HARs, and are de novo CNVs of HARs more common in cases than controls? And so de novo CNVs that involve HARs represent about a quarter of all of the de novo CNVs, but you see that they are 6 times as common in cases as they are in controls. And most of these CNVs are large. Most of them delete multiple genes, so they don't just delete a HAR. They delete exons as well. But nonetheless, it does suggest that there might be a signal there. And here's an example of a particularly intriguing HAR that's involved in a copy number variant. This is a duplication that's about 75,000 base pairs large. It occurs in this segment of the genome, and there's no genes in there. The nearest gene is this NR2F2, which is actually over here. It has an antisense transcript going in this direction. And so this is really just a duplication in the middle of nowhere. But then if you go to this chromosome capture technology, you can see that actually previously published experiments actually using a related technology called ChiaPet this red thing is the HAR. The HAR is in a large duplication, and that HAR bears the marks of it being an enhancer with this H3K4 monomethylation in fetal brain. And then, in fact, there are direct sequences that connect that HAR to this gene, NR2F2. And that gene NR2F2, otherwise known as CoopTF, encodes a transcription factor that's already been shown to be essential for the development of specific subtypes of interneurons in the cortex. In fact, you have to downregulate the gene in order to allow these interneurons to enter the cortex. And if you overexpress the gene, they fail to enter the cortex properly. And I don't know whether this duplication would cause higher levels of gene expression or lower levels of gene expression, but it would undoubtedly have an effect on the dose of the gene. So then to look for recessive mutations associated with HARS, we turn to our Middle Eastern cohort because we know that recessive mutations are about 10 times as common in people who are married to in the offspring of people where parents are cousins. And so we thought that this would be a population enriched for recessive mutations. And so for this, we looked at whole genome sequencing from 27 families. And then for the remaining 186 families, we basically developed a HARome panel analogous to an exome panel, a capture array that captures all of the or roughly all of the 2,700 HARs in the genome and sequenced it at very high coverage. And so what we found was indeed, if you look at rare homozygous mutations in HARs, they are significantly enriched in cases compared to controls by about 5%. And if you actually limit your attention to those HARs that bear marks of neuronal enhancers, in fact, that difference becomes even more significant. It's almost, but not quite, it's 1.75x, so 75% more common, suggesting again that about half of the HAR mutations, these recessive HAR mutations, are likely to contribute to risk. So the ASD probes show a higher rate of rare HAR variants. The excess is greater when you focus on the brain ones. And overall, it may contribute to risk in as many as 5% or so of the consanguineous families. And so we wanted to know, are these mutated HARs active? What's the effect of the mutation on the enhancer activity? What are the genes that are actually regulated by the HAR? And then do these mutations actually alter the activity of the enhancer or the HAR in vivo? Now, we didn't do all these experiments, but I'll show you an example where we've done a lot of the experiments and a couple of other examples of where the work is still in progress. So this is two families, one with 2 affected males, one with an affected female. They share autism spectrum disorders and intellectual disability. They share linkage to chromosome 7 in both families, and neither of these families has a particularly likely causative mutation anywhere in the exome. And then they both share a G to A mutation here in this HAR. So this is greatly magnified. The HAR is only about 200 base pairs long. And you can see that it's likely to be an enhancer. It's a DNA-sensitive site in fetal brain, which suggests that that DNA is open. It's also bears other marks of being active chromatin. And then, in fact, if we look, if we zoom out, here is that HAR corresponding to the black bar shown at a lower magnification, the HAR within that segment. And again, that is in the midst of an enhancer peak, suggesting it functions as an enhancer. And so you would naturally expect it would be an enhancer for the nearest gene, MYL10, which is expressed in muscle. But in fact, again, if you look at this chromatin capture data, it doesn't seem to regulate that gene. It seems to regulate a gene which is quite some distance away. And that gene is called CUX1. Now, CUX1 is a remarkable gene. It's the vertebrate ortholog of a Drosophila gene called CUT, which was described about 20 years ago as a dosage-sensitive regulator of dendrites of neurons. And so in the fly, neurons with complex dendrites have high levels of CUT expression. Here, high, you have complex spiky dendrites. Neurons with low levels of CUT expression have very simple dendrites. And if you take a neuron that normally has a complex dendrite and mutate CUT, the dendrite becomes simpler. It lacks all these little branches. Or if you take a neuron that normally has a relatively simple dendrite and you overexpress CUT, it makes the dendrite more complex. And so it's very sensitive to dosage. Either up or down changes its function dramatically. And the exact same thing is true in mammalian neurons for the ortholog CUX1, the target of this HAR. It's normally expressed in the upper layers of the cortex, which is fascinating because the upper layers are the most recently evolved evolutionarily. And it's also expressed in a subset of interneurons called reelin-positive interneurons. And in fact, it's essential for the development of both of those cell types. And if you deplete CUX1 from the mouse in experiments from Martin Nieto's lab, the dendrites become simpler, and if you overexpress it, the dendrites become more complex. So it has very analogous dosage-sensitive properties in the mouse. So Ryan then isolated the HAR and put it into an expression construct and showed that, in fact, that HAR enhances the expression of a gene consistent with it being an enhancer. That's the definition of an enhancer. And then he created the autism mutation and actually surprisingly found that that made the enhancer work even better. The enhancer was about 2 times stronger. And this is in a neuron-like cell line called N2A. And in fact, if you then take those N2A cells and you differentiate them into neurons using dominant negative rest, the enhancer activity goes up and the effect of the mutation goes up even higher. And you can see here the mutation changes the sorts of transcription factors that would bind to the HAR element. And just to make sure that this would also happen in vivo, he created a similar enhancer construct and connected it to green fluorescent protein. So wherever the gene would be expressed, you see green. And you see that normally it is an enhancer, and then you create the mutation and the brain glows brighter because the levels of gene expression are higher. And so we think that mutations like this can change the levels of gene expression or their pattern and might cause more subtle behavioral defects perhaps than coding region mutations. Here's another HAR mutation. This was a very deleterious looking one where you remove actually 5 base pairs from an enhancer element. And you can see that it's likely an enhancer based on the enhancer marks. And then again, this is a zoom out of where the HAR is. And again, you would expect it to regulate the nearest gene, but it doesn't. It regulates this gene, which is further away, called PTBP2. This is an essential neuronal splicing factor, which has been previously implicated in intellectual disability. And you can see that this particular HAR has enhancer activity. The mutation does not affect the enhancer activity in dividing cells, but when you differentiate the cells to neurons, you see that the enhancer activity is impaired by this particular mutation. And then here's one more example. This is a mutation shared by 2 affected boys in a family and not present in their siblings. It's a T to G mutation, again, in an enhancer, in a HAR that has the marks of an enhancer. Once again, it doesn't seem to regulate the closest gene, which is a long non-coding RNA, but in fact is connected to 2 nearby genes, one called TMEM161B and another MEF2C, which is, again, a known dosage-sensitive transcription factor essential for neurons, which has been previously implicated in gains or losses being associated with intellectual disability and autism spectrum. So we have about a dozen of these HAR mutations. Again, we don't think they're all causative, but we think a certain fraction of them are. There's another HAR that actually has 2 different mutations in 2 different families, again, in a region that has previously been implicated in dosage-sensitive intellectual disability. There's a HAR which inserts a half a dozen base pairs into an enhancer for CDKL5, which is, again, a known autism gene. So this is, again, very likely to be a causative mutation. And there are some others that, frankly, we're a little less sure about. So these ASD mutations seem to identify a subset of essential HARs. About 2,000 SSC families identify dozens of candidate HARs involved in these de novo CNV, though most of the CNV also will involve exons. The consanguineous families identify another dozen or so rare linked HAR SNVs. What we really need to do then is explore HAR variation in larger numbers of patients to see whether they contribute to autism spectrum disorders in the United States as well as in consanguineous families, to see whether they might cause other conditions as well. We're really only scratching the surface of 2,700 HARs, and we have possible relations for a couple of dozen of them. We have a lot we need to understand about what functional changes in expression are caused by the autism-associated mutations, are caused by the species differences in HAR sequence, and whether these patterns of enhancer or HAR activity might, again, enlighten us as to the anatomy of autism spectrum disorders. This just shows an example of a HAR, the human version and the mouse version. If you connect them up, you can see that they direct different patterns of gene activity, and that's really seems to be the basis of these HARs. And so we think that they might have again, different patterns of usage in evolution, as well as potential different changes after these very severe mutations. So just to conclude by acknowledging people, this is the somatic mosaic work. The work that I presented was really led by Ann Paduri and Alyssa DeGamma. Gilad and Shuyu, 2 great former students, did a lot of beautiful work that I didn't really have time to present in great detail. Elaine Lim has done the analysis of somatic mosaic mutations in the 6,000 autism samples. We have a great collaboration with Peter Park's lab, and we're very grateful to the New York Genome Center as well as other places for help with sequencing. And again, the support of the Simons Foundation is especially important for this Middle Eastern project. That was the HAR mutation work was done by Ryan Doan, the CNV0 work by Klaus Schmitz-Abe, Kyriakos Markianos, and then Ryan and Tim also contributed, and our many collaborators in the Middle East made it all possible. Thanks very much for your attention.